Hello, all students, the NEET aspirants. So, it is my effort to make you feel so comfortable to read in between the lines of the text by untwisting of all the knots of NCRT text. So, keep the textbook with you. How my delivery synchronizing with the text, that is very, very important. And finally, you are going to feel so comfortable, and whatever the things present in the text in different frames is going to be registered in your brain. So we will switch over to the topic, the biotechnology. So keep the textbook with you. In very first part, the Rene Descartes, 17th century naturalist, philosopher, mathematician. So from his time period onwards, all our human efforts with regard to the natural science directed to develop technology. The technology in turn meant for creature comforts for humans, values to human life. And whole approach of understanding natural phenomena, it became anthropocentric, that is the human-centered development. If we link this to the first topic, the living world, the societies indulged in anthropocentric view registered very limited progress in biological knowledge. That means the human-centered development. That's why biology remained as a natural history for the long time. If you link this sentence to the 11th class, the morphology of the flowering plants. Before experimental biology, more specifically physiology, become established as a part of the biology, naturalists described only biology. That's why biology remained as a natural history for the long time. That's why the biology emerged in 20th century, the modern biology. And offshoot of the modern biology, as per 20th century, it is biotechnology, whose products brought great qualitative improvement in case of the human life, whether food products or agricultural products or anything. It is everything, it is for human centered only. Even physics, chemistry contributed for technology, industry, engineering. Again, it is contributed for the human welfare. And in this topic, especially the whatever the things, the plants and animals, it is related to production of what it is the food. And in this, we are going to have the biotechnological principles and process, and finally going to have the biotechnological applications. Next, with regard to the scientists Herbert Boyer and Stanley Cohen. Herbert Boyer, born in Western Pennsylvania. Why there is a need to stress the Western Pennsylvania? It is a remote place. Born in such a remote place, and greatest discovery was attributable to Herbert Boyer, who discovered couple of restriction enzymes with useful properties. That means these enzymes cleave the DNA in a particular fashion. That means at the double stranded ends, there is a single stranded ends present. At a clipped ends, having what it is single stranded sticky ends, the cohesive ends. And the cohesive ends readily pasted as a precise exercise. So cleaved the DNA and pasted the DNA fragments. So readily pasted as a precise exercise, what we call it is the DNA splicing. Next, who met Stanley Cohen, a Stanford scientist who had been observing ringlets of the DNA present in cytoplasm of the bacteria. That is, in addition to the genomic DNA, in addition to the chromosome, bacterial chromosome, circular, naked, double-stranded ringlets of the DNA present, what we call it is the plasmid. And the plasmid, it can replicate independently of the coding strand. Irrespective of the chromosome, it can replicate that is independently in case of the cell. So who simply isolated the plasmid and reinserted the plasmid into bacterial cells. So simply unaltered plasmid introduced into bacterial cell. who developed the technology of isolation and transferring the plasmid to the bacterial cell. Next, combining the process, that is Herbert Boyer, DNA splicing, cutting and pasting, and next process, isolation of plasmid and transfer to bacterial cell. So combining the process of Herbert Boyer and Stanley Cohen, recombine the DNA in a desired configurations introduced into bacterial cell. So unaltered plasmid transferred into the cell by Stanley Cohen. Altered plasmid with a desired configuration introduced into the bacterial cell by Herbert Boyer and Stanley Cohen. That step acts as a great thing, great breakthrough in case of the biotechnology, because it acts as a manufacturing plant for specific protein. And total biotechnology is going to depend upon this, especially this step, that is recombining the desired configurations and introduced into the bacterial cell that act as a manufacturing plant for a specific protein. That is a great breakthrough in case of biotechnology. Next, we switch over to the real topic, that is biotechnological process principles and process. In very first paragraph, the living organisms, its engine to produce the products and process, that is the biotechnology. That is technology, living organisms, and we are getting what? The products. And traditional biotechnology, as René Descartes' period, that is ancient biotechnology. And in traditional biotechnology, curd making, bread making, wine making are all nothing but it is biotechnology, because it is micromediated. So all micromediated bread making, wine and curd all involve nothing but it is biotechnology. Next, but biotechnology used in restricted sense today, that is the current day, that is the production of the curd, bread, wine by using genetically modified organism. What do you call it? Is biotechnology. So we are producing those things in large scale by using genetically modified organism. Next, biotechnology also includes in vitro fertilization that leads to test tube baby, DNA vaccines, hepatitis B vaccine like that, and synthesis of the DNA and using it, and defective gene is replaced. So all it is included in case of biotechnology. Next, European Federation of Biotechnology, EFB, or European Federation of EFBT, European Federation of Biotechnology, encompass, that is combining traditional view as well as modern molecular view, that is emphasis. So traditional view as well as modern molecular biology, they gave the definition for biotechnology. Integration of natural science, cells, parts, molecular analogues, that means molecules that mimic the structure and function, that what we call molecular analogues, that used in production as well as for the process, and different products are useful to the human beings. And this definition in a broad way, exploitation of the microbes, their properties, cell constituents at industrial level to generate useful products for human beings is nothing but biotechnology. Next, biotechnology based on number of branches, but the two core techniques enable the birth of the modern biotechnology. It is genetic engineering and bioprocess engineering. Genetic engineering, nothing but manipulation of the gene, tailoring of the gene, alteration the chemistry of the DNA and RNA, nothing but changing the genotype. Change in the genotype means change in the phenotype. Next, bioprocess engineering. So as to produce the product from the microbe, there must be some congenial condition, favorable condition. That means it is free from other microbes, uncontaminated atmosphere. So we have to create such conditions. So ambience for sterility is created through chemically engineered process. So we are looking for the ambience for sterility to enable the growth of the particular microbe, prokaryotic cell or eukaryotic cell, in uncontaminated atmosphere for large scale production, large biomass, high biotechnological product. So any organism, microbe is going to yield the product in large scale in uncontaminated atmosphere. So we have to create such conditions, that is sterile condition, what we call bioprocess engineering, and these conditions which are available in case of tissue culture. Next paragraph information. What is the concept behind it in development of the principles of genetic engineering? Suppose if we take the sexual reproduction, the meiosis, the Pachytene stage, at this, the exchange between non-sister chromatids, what we call the crossing over, results in recombining the genes, that results in variations. So sexual reproduction is advantageous than asexual reproduction. New genetic setup, new combination of genetic setup is going to be created, leads to variation, which may be useful to the organism or it is total population. And if you take asexual reproduction, genetic integrity is maintained in contrast to sexual reproduction. Next, in natural hybridization in plants and animals, that is traditional hybridization, combining the desirable as well as undesirable genes is possible. Not only desirable genes, undesirable genes also possible. So inclusion and multiplication of desirable as well as undesirable genes occur in case of traditional hybridization. So suppose if we take A and B plants, the hybridization between A and B plants is going to give C, the hybrid. The hybrid is not always necessary to combine the desirable characters only. It also with undesirable character. Such extravagancy, such abnormality, such limitation is overcome by means of genetic engineering. That is gene transfer technology, recombinant DNA technology, gene cloning. So you can overcome with this. That means we can transfer the gene or set of desirable genes into target organism without inclusion of the undesirable genes. Next, we can transfer a piece of DNA into alien organism, it would not multiply. That means we introduce into cytoplasm. If you introduce a piece of DNA into alien organism, it would not multiply. Suppose it is assertion. The reason would be, unless it becomes the part of the genome, it would not multiply. The genome, the bacterial chromosome having DNA, DNA having ori. So if you link any foreign DNA to ori, it can multiply and produce indefinite number of copies. What do you call the gene cloning? Or getting template of what it is the DNA in indefinite number. Suppose you are inserting the gene in ori, we are not going to get. We have to link the gene to ori. Suppose if you inserted the gene into ori, the ori is going to be deleted. So we are linking the foreign gene to ori. It can produce indefinite number of copies. What do you call it is gene cloning, or getting multiple copies of template DNA. Next. In next reading, the construction of first recombinant DNA by Herbert Boyer and Stanley Cohen. Only there are the 4 or 5 points in the total paragraph. The gene encoding for antibiotic resistance was cut out from the plasmid of Salmonella typhimurium, not from the genome, it is from plasmid, due to restriction endonucleases. The restriction endonucleases are molecular scissors, molecular scalpels, which cleave the DNA in a particular fashion, results in single-stranded sticky ends. So gene encoding for antibiotic resistance, it should be expressed. It should be expressed in case of what it is particular host. So it should be transferred. It requires a vector, a Ferrier molecule. And by using the same restriction enzyme, who cut the plasmid of Salmonella typhimurium, that is native plasmid. In this also sticky ends are formed. So the sticky ends in both, which is ligated by using DNA ligase, and ligation or annealing from end to end, from one end to what it is other end. Such end-to-end ligation results in a circular, naked, double-stranded DNA molecule. So, DNA, it is a vector. It is the foreign DNA gene encoding for antibiotic resistant. Now, what do you call it is? Recombinant vector or recombinant DNA or chimeric DNA. So, in order to express the gene encoding for antibiotic resistance, it should be transferred to host cell. The host cell is going to be Escherichia coli. So, we isolated the gene encoding for antibiotic resistance from Salmonella typhimurium, and for a carrier, we isolated the plasmid and cut the plasmid from Salmonella typhimurium. In order to express the gene encoding for antibiotic resistance, we are transferring to Escherichia coli. It is expressing system. So, gene encoding for antibiotic resistance multiply in case of the host cell, independent of the bacterial chromosome, but dependent on deoxynucleotide DNA polymerases of the host. So, DNA dependent DNA formation here, and we are getting multiple copies of the gene encoding for antibiotic resistance. What do you call? Cloning of antibiotic resistant genes in E. coli. So, in this, the question mode, the gene encoding for antibiotic resistance was cut out from plasmid of Salmonella typhimurium. The gene carrier is from Salmonella typhimurium, and expressing system, the gene encoding for antibiotic resistance expressed in, that is bacterial cell, that is Escherichia coli. Escherichia coli and Salmonella are similar because both are, it is prokaryote. Next, you have to modify the genetically modify an organism. There are three basic steps. First, you have to identify DNA with a desired gene. Next, you have to introduce into organism, and maintenance in the organism, and subsequently transferred to the progeny cell. That is important. Maintenance in the organism is very, very important. What is the maintenance in the organism? So, maintenance in the organism require, in culture medium, we must have the antibiotics, because the host cell always tend to lose the foreign particle, that is the DNA, other DNA. So, it should be retained in the cell. So, it must have the pressure externally, that is in the form of antibiotics. So, in order to bear this pressure, it must have antibiotic resistance genes, antibiotic resistant genes in case of the vector. So, identify DNA with the desired gene, introduce into organism, maintenance, and subsequently transferred to the progeny cells. Next, what are the various tools in recombinant DNA technology? We require restriction endonucleases, DNA polymerases, DNA ligases, vectors, and host cell. These are the vector for successful accomplishment of what it is recombinant DNA technology. Next, restriction enzyme. The name got because of, as it restricts the multiplication of the viruses inside the bacterial cell. So, bacterial cell developed a mechanism to prevent the growth of viruses, to restrict the multiplication of the viruses. What is the modification enzyme system in case of Escherichia coli? The one enzyme, the modified enzyme, adds methyl groups to the host DNA, and one enzyme cleaves the incoming viral DNA. So, whatever the enzyme which cleaves the incoming viral DNA and it restricts its multiplication, what do you call it is restriction endonuclease. And other enzyme which adds methyl groups to the host DNA, bacterial DNA, it is called methylase. So, host DNA got well protected. Why? Because of addition of methylase, not recognized by restriction enzyme. Why viral DNA got cleaved? Because it is not methylated, recognized by restriction enzyme. It cleaves the viral DNA. As it restricts the viral multiplication, the name it is called restriction endonucleases. Next, the first restriction enzyme discovered was HIND2. The 2 refers to order of isolation. First discovered, but isolated the second from that Haemophilus influenza bacteria. Next, the HIND2 having special recognition sequence GCT AGC. So in GCT and ACG GCT. So in this we are going to get what it is the blunt ends. 6 base pairs, that is the 12 nitrogen bases, is it recognition sequence. So what is the recognition sequence? The number it is the 12 nitrogen bases, that is 6 base pair. What do you call recognition sequence for HIND2? Next, so it is characterized 5 years later to this discovery. So order of isolation second, but discovery first. Next, ECOR1, the restriction enzyme, it was extracted from Escherichia coli RY30. The E refers to, in case of ECOR1, E refers to the genus Escherichia. CO refers to species, that is the coli. R refers to strain. Roman 1 refers to order of isolation from that Escherichia coli, that is RY30. And ECOR1 is a restriction enzyme extracted from the bacteria. More than 900 restriction enzymes which are extracted from over 230 strains of the bacteria, and restriction enzymes which are absent in case of eukaryote. And these enzymes belongs to large class of enzymes. What do you call it is nucleases. And these nucleases belongs to the hydrolases, that is the third class according to IUB system. The restriction enzyme which belongs to nucleases, which is the large class of enzyme, and that belongs to it is hydrolases. And in case of the nucleases, it is exonuclease and endonuclease, the two types. Exonuclease, the word itself conveying its meaning. It acts outside the DNA, it must have the free ends. So it cleaves the DNA into fragments. The exonuclease do not act on circular plasmid because circular plasmid do not have the free ends. Next, the endonuclease. Endonuclease act within the DNA. What do you call it is endonuclease. So all restriction endonucleases are endonucleases, but all endonucleases are not what it is restriction enzyme, because endonuclease whose cleavage is not specific, it is random, but restriction endonuclease is specific at particular sites. Next, the restriction endonuclease, first it inspects the length of the DNA sequence. Next, it binds to the DNA. Next, it cleaves the DNA in a particular fashion. That is, first it inspects the length of the DNA sequence, that is 6 base pair, and find the recognition sequence, that is the palindromic sequence, and binds to it and read and cleave the DNA in case of particular fashion. The palindromic sequence, if you take the word Malayalam, it reads the same both forward as well as backward direction. But in contrast to this, the palindromes in case of the nucleic acid read same in forward and backward direction. One orientation of the reading kept same, 5-3 GAA TTC. In this also 5 and GAA TTC. That is reading same both forward and backward, when orientation of reading kept same, 5-3 5-3, otherwise 3-5 3-5 GAA TTC. And this ECOR1, it breaks it identifies GAA TTC. It breaks between G and A on one strand and other strand also G and A. What do you call such a cleavage? Symmetric cleavage in between the same nitrogen bases, that is the purines. adenine, then guanine, adenine, guanine, adenine. So it breaks between purine, 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 purine. So symmetric cleavage of phosphodiester bond on both strand at a different location just away from the mid of what is the palindrome results in single stranded sticky end at the end of double strand. What do you call it is overhanging ends, protruding ends, sticky ends, cohesive ends. So we can frame the number of questions here. So what is the length of the palindrome as it is having 12 nitrogen bases? So 20 nitrogen base having 34 angstrom. 12 nitrogen base is going to be 20 point something angstrom. So in this palindrome, how many hydrogen bonds which are still intact? 6 hydrogen bonds are intact. GC, 6 hydrogen bonds are intact. How many hydrogen bonds got disturbed? 8 hydrogen bonds got disturbed. AATT. 8 hydrogen bonds got disturbed. How many double bonds it got disturbed? 4 double bonds got disturbed. How many triple bonds are intact? 2 triple bonds which are intact. How many phosphodiester bonds broken? The 2. How many carbon nitrogen rings are there? As it is having 12 nitrogen bases GAA, TTC. So 6 purines, 6 pyrimidines. As purines are dicyclic, 6 into 2, 12. And pyrimidines are monocyclic, that is the 6. So total it is 18 carbon nitrogen rings in case of recognition sequence, that is GAA, TTC. Suppose if you take a fragment of the DNA having 4 recognition sequence GAA, TTC at 4 places, you are going to get how many fragments? 5 fragments you are going to get. In this 5 fragment, last 2 fragments is going to be single strand at the end of the double strand. And remaining 3 fragment, single strand and either side of the double strand. So totally you are going to get the 5 fragment. The principality is n plus 1 in a linear segment. Suppose if you take the circular plasmid having sequences at 4 places, the 4 sequences are there, GATTs at the 4 times. You are going to get the 4 fragments only, n is equal to n. What is the nature of the fragment? Single stranded on either side of the double strand. So in case of the linear, n plus 1, we are going to get 5. In case of the circular, we are going to get n is equal to n, the 4 fragments only. In case of the linear, last 2 fragments, the single strand at the end of the double strand. And 3 fragments are going to be single strand on either side of the double strand. In circular, single stranded ends on either side of the double strand. So n plus 1, n is equal to n. Next.